All living organisms require accurate segregation of their genetic material. Here, we investigated this fundamental process in a bacterium which is associated to a marine nematode, namely Laxus honestus, which belongs to the family of Stilbonematinae. Freshly isolated worms appear white under incident light, because the nematode maintains a virtually pure culture of a sulfur-storing gammaproteobacterium Candidatus theonesti on its cuticle. These rod-shaped symbiont cells ensheath the whole animal with a dense monolayer. Although Stilbonematinae regularly occur in tropical and temperate shallow water sediment, no Candidatus theosymbiont has been isolated so far. If we zoom onto a bacterial cell, we find it attached with one pole to the cuticle so that its long axis is perpendicular to the animal surface. To stay attached throughout the generations, symbiont cells grow in width and divide longitudinally, which was shown in previous studies. We hypothesized that the uninterrupted host attachment would affect bacterial chromosome dynamics and set out to localize specific chromosomal loci and putative DNA segregation proteins. It was shown in model bacteria that chromosome segregation starts with the separation of the replication origin, then the bulk chromosome is segregated, and finally the region where replication terminates is separated. In some bacteria, the three-component ParABS system drives or assists ORI translocation. It consists of a centromere site on the DNA, shown in red here, ParS, a centromere binding protein, ParB, and a Walker type A ATPase, ParA. In short, this is how it works. ParB binds specifically to ParS, and DNA-bound ParB interacts with ParA, which is bound nonspecific all along the chromosome, and relocates by this interaction, presumably via a unique diffusion ratchet type mechanism, the emerging sister chromosomes to opposite cell house before division. To determine the ploidy of the nematode symbiont Candidatus theonesti and the localization pattern of its origin, we fixed it and subjected it to DNA fluorescence in situ hybridization. The histogram in Figure 1a shows that the vast majority of cells contained either one or two fluorescent foci. If we assume that one fluorescent focus equals one ORI copy, we conclude that the nematode symbiont is monoploid. This conclusion was also supported by counting the number of fluorescent foci obtained with an anti-ParB antibody shown in Figure 1d. After determining Candidatus theonesti's ploidy, we analyzed the ORI localization pattern throughout the symbiont life cycle. In non-dividing cells displaying only one focus, it occupies a near mid-cell position. And before the onset of septation, a process concomitant with symbiont cell growth, the sister ORI regions are segregated diagonally, which is along the short but also along the long axis. A very similar localization pattern was observed for ParB, a near mid-cell position in non-dividing cells and then migration towards opposite cell house diagonally. We concluded that Candidatus theonesti is monoploid and that its sister ORI segregate diagonally, which is along the short and the long axis before septation starts, and that ParB recapitulates the ORI localization. To determine the localization of the terminus of replication, we applied DNA fish targeting a chromosomal region containing the predicted diff site. Along the progressing cell cycle, cis-tertiary migrate first away from each other diagonally before Candidatus theonesti starts dividing. Then, as septation begins, they localize medially at the divisome leading edges. Finally, as septation progresses, each migrates towards midcell. Panel D and E show this migration towards the division plane and towards the cell center when approaching the end of the cell cycle. In non-dividing cells, the tear position on the long axis resembled that of ori, whereas on the short axis, tear appeared closer to the cell envelope than ori. We concluded that the symbiont chromosome is transversally organized. Summarizing, cysteid tear migrate away from each other diagonally before Candidatus theonesti division starts, and as septation begins, they localize medially at the divisome leading edges and migrate concomitant with ingression towards midcell. And finally, this is our model for the chromosome segregation of Candidatus theonesti. The bidimensional segregation mode of ori and tear, which is diagonally toward opposite cell halves before division, and the reapproachment of tear during the prolonged septation period, maintains the symbiont chromosome orientation toward the animal surface transgenerationally. We hypothesized that it might be advantageous to permanently position genetic loci mediating host interaction in the vicinity of the host and to confine loci involved in environmental response to the distal cell half. From a chromosome-centric evolutionary perspective, even the peculiar reproductive mode of the symbiont might have evolved from the need to maintain a specific chromosome configuration.